I'm Saloni Kernwal,
I'm Brian Arceo.
And I'm Matt Lohr.
and today we're going to be presenting about microalgae and wastewater remediation. Algae are a class of photosynthetic organisms that are found in both marine and fresh water, making it an integral part of the ecosystem. They make up 70% of the world's biomass. Algae thrive on carbon dioxide with resources and nutrients such as sunlight, nitrate, and phosphate. Carbon is responsible for algae metabolism and production of lipids and carbohydrates. We work with Neochloris oleoabundans, a strain of algae that was grown in BG11 media, to measure biomass density followed by lipid extraction.
During the growth period of 8 days, the photobioreactor was kept at 25 degrees Celsius with an alternate exposure to light, 12 hours on, 12 hours off. Biomass density was measured by cell count, spectrophotometry, and gravimetric analysis. Hemocytometer was used when taking the cell count of the culture. Absorbance was measured at 680 nanometers, corresponding to chlorophyll A, and 750 nanometers, and gravimetric analysis with a glass fiber membrane to obtain dry and wet weight. For lipid extraction, 100 milliliters of grown cell culture was concentrated by centrifugation and resuspended in methanol. Lipids were extracted by heating at 60 degrees Celsius for 30 minutes, and solids were removed by filtering extract with a glass fiber membrane. Methanol and acetone were added to decolorize the cell. Solvent was removed by the filtrate by evaporating at 60 degrees Celsius. The residue was then resuspended into a vial and evaporated once more to obtain total lipid extracted.
We observed the growth of Neochloris oleoabundans in pistachio water over 17 days. According to the graph, the initial concentration of nitrogen and phosphate showed a growth phase of NO. The growth rate slowly declined with nutrient deficiency. Based on the end result, nitrogen and phosphate need to be further analyzed and experimented on to find advantages in pistachio water. The purpose of the experiment was to show the environmental and financial potential algae has. It grows fast, consumes carbon dioxide, purified wastewater, and are used as an energy source. Algae are used in nutraceuticals, which are food that contains these health-giving additives and have medicinal benefits. Furthermore, algae contains high biofuel yields for potential services.
Now let's move on to our next presentation about tiny flies in research. Drosophila melanogaster, powerful research tool. Drosophila melanogaster, also known as fruit flies, are commonly used model organisms in biomedical research. Flies are genetically diverse, simple to work with, and have a short generation time period. They are used to study disciplines ranging from fundamental genetics to the development of tissues and organs. When compared to humans, Drosophila melanogaster chromosomes comes in pairs with one set of sex chromosome and 3 pairs of autosomes.
Similarities between humans and flies are their cell biology. Both have similar regulation of gene expression, membrane trafficking, cytoskeleton, nervous system, cell signaling, and cell death. In contrast, flies have much more simpler circulatory systems and cognitive processes. Only one duplicate gene exists in flies, while there is repetition in humans. Now let's examine how fruit flies can be used as model organism in biomedical research. Reverse genetics help with identifying genes purely on the basis of their position in the genome, with no knowledge whatsoever of the gene product. This process helps us find clues on how to counteract human mitochondrial diseases. As for forward genetic, it is a molecular genetic approach of determining the genetic basis responsible for a phenotype. This process helps us assess a specific homologue that can exist in human diseases. Let us look at one of those neurological diseases, Parkinson's disease.
Parkinson's disease is a neurodegenerative disease caused by degeneration of dopaminergic neurons in the substantia nigra of the brain. Mutations in familial genes that have been linked to Parkinson's disease include alpha-synuclein, Parkin, and PINK1. Homologs of familial genes Parkin and PINK1 exist in Drosophila melanogaster. Mutations in Parkin and PARK1 display Parkinson's disease phenotypes comparable to humans. This allows Drosophila melanogaster to be used as models to screen potential therapeutics that suppress neurodegeneration.
In the Feeney and Bender fly model, they used the mutation of alpha-synuclein on wild-type and mutant form. They concluded that the mutation gave progressive motor impairment, and it was done when the fruit flies had an absence of tyrosine hydroxylase chemoreactive neurons in the central brain. In addition, they used the eye as a model to look for retinal degeneration. In the picture, the control had a well-preserved retina when it was 30 days old. As for the alpha-synuclein expressing fly, there were signs of retinal degeneration. And that concludes our presentation on how algae and Drosophila melanogaster can be fuels for the future of science. Thank you.